Hi, and welcome to For Whom the Cell Tolls. It's been a while, but I'm your host, Keenan. Today, Scout Pomeranian is not joining me, but one of my great coworkers is Katie Peterson. So she works at Gustavus Adolphus College with me, and we both teach undergraduate biology. Oh yeah, and so Katie, I'll allow you to kind of introduce yourself and kind of go over your research and what's exciting that that you study.
Okay, great. Thank you, Keenan, for having me on. So again, my name is Katie Peterson, and I am actually a Gustavus alum, so it's been really exciting to be back at Gustavus in a different capacity as an instructor for this year. My research is kind of all over the board, I would say, but generally falls into evolutionary biology and kind of organismal biology. So I'm really interested in how creatures that we don't often think about are experiencing and kind of navigating the landscape around us. And so I do that kind of using island biogeography and a naturally fragmented system to hopefully extrapolate maybe what will happen potentially or what could happen to improve restoration efforts because the world around us is starting to kind of look and mirror the landscape that I study in southern Idaho.
Awesome. And this will be a very good, fun chance to kind of have a discussion about how, although I research something very molecular, the listeners of this podcast know that I'm very, fond of evolution ecology and, looking into, larger systems and seeing where the kind of that territory kind of melts together, and that's super exciting and fun. So this episode will kind of detail Katie and I's journey a little bit, how our research is, super fun when it collides, how we ended up actually kind of along a teaching path, which is, I always think, really exciting. And then we'll address some student questions that we got from Gustavus students, so that'll be good. So for me, I can just start and, my research is pretty much the same boring stuff that we've talked about this whole time, but for those that are new, listening from Gustavus, I mainly take, basically sequence DNA from tumors in patients, and that's what we do at Mayo Clinic, and I make sure that, or I at least designate what kind of therapies are going to be the best treatment options based on the genetics in the tumor. This is how we can personalize medicine and basically further patient progress and survival better than just a single therapy that is just going to, hopefully work for everybody, but in all likelihood not work for everybody. This way we can save patients a lot of hardship and improve, progression rates and all kinds of stuff. I also work really closely doing a lot of cellular mechanism studies where I go kind of deeper into the cells and mess with things and see if that makes them angrier, sadder, depending on the cancer type and cell, and see how we can take advantage of those mechanisms in therapy. So one of the exciting kind of crossovers that I always introduce in a lot of my classes is that when you have a tumor, it is very much kind of like an invasive species. It has its own separate genetics, it's not behaving correctly in that environment, and it is kind of wreaking havoc. And I can't remember what the rule is, you can help me on this, Katie. There's some, I think there's a stipulation in ecology that says invasive species are almost unstoppable or something, or you can never eliminate them 100%.
Yeah, I mean, in some cases, partially just that kind of release, right? There's nothing that's kind of keeping them in check, right. Is one thing. And generally their reproduction is also quite, what's the word? They're able to reproduce quickly too, so I guess that's kind of the other side of it too. It's trying to maintain them.
Yeah. No, and it's super fun to watch. Well, no, it's not fun. Sorry, I always do that on this podcast where I'm interested in something and I say oh, this is fun. No, it's not. So it makes it tough, and especially when you put those two sciences together, you can see kind of the daunting challenge both types of science are up against whenever you are looking either at something that's invasive or an environment that is going to change and make a better opportunity for something that can survive generally, for example, versus something that can only survive specifically. And then Katie, you kind of work on generalist specifics, right?
Yep. Yeah, so for my research, I study both plants and also specific groups of spiders because I'm interested in the community level of things. And so the spiders that I'm talking about right now are the jumping spiders, and so they're the really cute ones that I'm sure you've all heard of or seen the really funny videos of them. doing their kind of weird dancing behaviors. But the jumping spiders, in my research, I use them to kind of be a proxy for something that would be considered a generalist. So they're kind of like the cats of the spider world, and they're actively seeking prey. They have really good eyesight, and they're walking around. So I'd say they're, a generalist in that they can find prey that, they're not relying on anything else. And the other group of spiders that I study are the crab spiders. And so they lie in wait for things to come to them. So they're more of an ambush predator. And so they're relying essentially on the vegetation around them to be able to camouflage onto them. So crab spiders can actually change colors, which is crazy. And so these crab spiders kind of are specialized in that their ability to essentially get prey relies on the vegetation around them.
Awesome. And one thing that always interested me about that, and I wondered if you ever got this question, is do people ever ask, like, well, is the generalist species just better then? Is that like, do you ever get that? And it's like, and then how it is somebody simple like me asking that question, how would you like help them understand?
No, I mean, I think like evolutionarily, if you're thinking about this, as soon as you start down a path where you're specializing, your chances of extinction, or essentially your life getting harder, kind of increases, right? Because you're getting more tied to specific resources, whatever they are that you're specializing on. And so, a benefit would be, now they're able to camouflage, so they have this kind of specialized response, to this, environment around them. But then, like where I work, I have pretty barren lava flows where there's not really vegetation. And so the cost of that kind of specialization for those crab spiders is they're not going to be successful in that other landscape. And so I think just in general, anytime you start really focusing and specializing on things and creating those really important ties, it can, in the long run, it might not work very well in a changing landscape.
Because it's almost like a risk reward, right? Like if you specialize into a niche, then you're going to reap all those benefits. But if the niche goes away, then you're completely out of luck. Yeah. It sounds like the high risk, high reward type of thinking that usually gets me into trouble with just anything. Like, can I make this exit, or can I make this red light, even when the reward is nothing? So, but yeah, I mean, that's something that, and I've shared this with Katie before, how excited I always get at the evolution ecology section, even though I'm a molecular person. I do have so many students, well, we both probably have students that both reject both of our arenas, and they're like, get me out of here. Like we were talking about students that we've both had, I think, students that are like, I just cannot stand cell biology. It's so boring. They're just cells. And then I have students that are like, ecology just troubles me. I don't like Hardy Weinberg. I'm scared. This is terrible. And I'm always just like, no, it's all fun. Like, this is so good.
Yeah. I mean, it's hard because obviously we chose our career paths because we're passionate about them. And it's hard for us to understand sometimes why, the people we're talking to are also not super excited about, what we are. And I always think that's fun and interesting because it really pushes you outside of your box to kind of figure out how you can relate and kind of make them care, about what you're doing.
No, and I always, try and look back to when I was an undergrad, and like when like the light bulb would sometimes go off for me was, when I'd hear like put something together that I was like, oh my God, is that true? And I think, hearing your work was so exciting for me because I was like, oh my God, like you'll actually watch those species go away each time one of those areas essentially shifts toward the changing climate. And have you actually had certain study spots, for example, that you've studied for years and then the next summer maybe they're gone? Or
no, so for my work I did 3 field seasons in the summer. 2 of them are ones where I was actually going into the islands of vegetation, so the kapukas that are surrounded by lava. So I have like 2 seasons worth of data, and there were a few kapukas that I went into both years because I wanted to get kind of that comparison, and I didn't see anything striking in that year difference. But one of the things, going back to when you're talking about like invasive species, something that's interesting I realized I never said. So I work at Craters of the Moon National Monument and Preserve in southern Idaho. So if you've never heard of it, Google it. It's a crazy landscape. Reminds me of The Martian. And so what's interesting is researchers in the past were looking at plant communities down there as well and trying to look at, invasive species and these islands that I go into. Some of them are more isolated than others. And they're actually finding that the kapukas that were closer to the railroads had a lot more invasive species, which makes sense, but until you're actually out there, like, kind of seeing the landscape and really putting that information together, it's not something that, you know, you would normally think about in this landscape that has lava all around, that these, you know, invasive quote-unquote species are still able to disperse into these pretty isolated systems, just because of the proximity to the railroad down there. So I don't doubt over time there's going to be changes there. And because the lava erupts there roughly every 2,000 years, like, you know, every 2,000 years there's some sort of change in that system that's applying pressure, you know.
Right.
Lava's flowing around, that's going to increase the heat, and, you know, things will burn. So yeah, there's constant changes, but it's on this time scale that we're not able to see.
Okay. No, and that makes sense. I didn't realize it was that easy to just transport, like, it's just seeds or something flying off the train and dispersing.
Mm-hmm.
Oh my God.
Mm-hmm. Mm-hmm. Yeah.
It's kind of...
plants are amazing.
It's kind of, they're pretty resilient. I remember when I was studying up for organismal, because we're teaching kind of in a similar, same class, I had to study my plants up again, and I was like, God, these things are really, like, resilient and, like, really advanced, and they have their own immune systems, and they're super fun. The train spreading reminds me of COVID-19 right now, spreading, basically, and just being like, zip, like, there you go. Because we're still walking Scout all the time, and sometimes, like, other people are walking their dog, and I was like, yep, we gotta go all the way over to the other side. I'm social distancing 30 feet, because you can't pet my dog. And I was actually joking with my my molecular class that a couple children tried to come up and pet Scout because she's cute. And I was like, no, shoe disease bags, carriers, get away. Like, don't pet this dog. But she was okay. She'll be fine. She gets so many walks now, so it was good. But yeah, so I always like to relate and, like, learn more about ecology because it's in evolution directly, because there are so many mechanisms that we never get to in introduction, and thus the ones that I usually learn the best are the introduction ones, and then I just apply those. But learning from you has been, like, really fun because I get to see just how quickly niche and environmental changes can change the environment on its head for these specialist type cells in a lot of cases. For me, when I'm studying something like cancer, and say, no wonder these certain like stromal cells that fight the tumor just go away when the tumor basically eats all their food around the area, or, like, kills off all their protectors. And we do something similar, and I've had episodes on this about CAR T and how CAR T cells are essentially supercharged genetic immune cells that go in and hunt the cancer. And I was thinking before this podcast, we were kind of talking about it in another conversation I was having, I was like, they're really hard to breed in captivity, these T cells that we're, like, modulating. And I was like, that's kind of the thing I was thinking of, and then, like, re-releasing and hoping that we can modulate the ecology of the environment. And, like, it rarely works, obviously. We're usually not in control of the environment as much as we'd like to be. So it was definitely interesting. Well, thank you so much for sharing. Yeah, this is fun. I think everybody on this podcast already knows what I do, so I won't bore everybody. And you can always Google and listen to the other episodes if you want. Because the other half of this podcast was kind of going to be, like, kind of an open session for us to talk about biology education, and kind of what some questions that our students had. So do you want to, I'm trying to think, we could first address the, one of the big questions that we get since we teach at kind of a smaller college is, where do you strike the balance of getting up and doing a pure lecture versus how many times do you break things up with different activities, for example? And the two of us, we have different strategies, but I think we probably almost exist on the same percentage. I hope. I hope I don't lecture too much. I feel like I do sometimes, so it's okay. I talk a lot. We're on a podcast, so it has to go somewhere into the air, basically.
Yeah, I think for me, I really try to think back to my coursework that I've had. I mean, even in graduate school too, kind of what professors were doing that made their courses really engaging and made me want to prepare ahead of time and be, participate fully in class. And those were the professors that definitely had kind of that balance point between not just, like, word vomiting towards me all the time, but also, doing activities. And I think it also provides a way for students to learn kind of in this group setting, but then also, I think, like you're saying, like the small group work gives students a chance to kind of build that confidence. So sometimes I'll, have a worksheet or questions up on the board, and I'll kind of have some breakout time because I want them to talk amongst each other. And then I bring it back in because I'm hoping the chance for them to kind of talk with a few folks around them, either, to get on the same page or, further explain something that maybe have a little bit better grasp on than the person next to them. And then, maybe that person, took something else away that they could, teach the other one. And then my hope is that, when we come back together, that kind of confidence boost of like, oh yeah, like we did kind of understand the same thing, allows people to participate more in that full lecture. Because I know it is kind of scary, and at times when you're in a room of like 40 folks to necessarily speak up. And so I think for me, finding the balance between lecture, because some of the stuff really, it is just kind of lecturing in my head is like the way that I learned it. And so I think that that's kind of a good base, but I really think the other experiences are necessary to not just get the information in one out here and out the other, but actually keep it in there, by engaging in those hands-on or whatever sort of experiences we're kind of concocting during our lecture times.
Yeah, and I definitely agree with like that time with like getting people together. I think it's definitely different in a bigger class. Like I didn't know that the Gustavus like intro would be only 35 students per group, and so I think a lot of the time some people were puzzled, like, wait, what? Like, I thought this was going to be a big, massive class. But having even just like a small conversation, you might see that somebody perceives something different, and you can perceive like a new association with something that maybe both of you are encountering for the first time, but seeing it from two different directions may make you think like, I guess I never really considered it that way, but maybe this is more disease-based, or maybe this is more like macro-based. Maybe this is better to look at as a large system. Or, because I did have a lot of, everybody has a lot of students that are really interested in healthcare. And so I try to relate it to that, but at the same time, I also don't want to send people into too dark of a black box of like, this is complicated and that's why it's exciting. And I think I struggle with that sometimes in the molecular class, where I'm just like, this is super exciting because it's super complicated and we're still figuring out, and that's exciting, but that also doesn't give the amount of wonder that I do want to have with some of this stuff. Because to me, a lot of that is like, hey, we don't know how this works, for example. Like, this is super exciting. It's terrifying that we don't know how it's terrifying that we don't know how anesthetic works, for example. Like, even just the simplest things. But like, the more that we find out about epigenetics, or the more that we find out about how the world is reacting to the changes that we're putting it through, which is probably twice as scary, which doesn't make me feel good ever. I remember, I think one of the biggest interdisciplinary things with climate change that I ever encountered was that when the Arctic unfreezes, viruses are going to come out of the peat that are still just DNA pieces, and they're going to start infecting things that they haven't infected for 1,000 years. No immune system is ready for them, like all this terrible stuff. And I was just like, oh shoot, okay, this is another way that we can have our morning coffee and just be terrified again. But right now,
I think that's the hard thing too with the content is like, there's so much like I want to foster curiosity in that you're saying like, oh yeah, there's all the stuff we don't know. And I think it's so crazy sometimes the stuff we don't know of like, well, it's it's written this way in a textbook, or we're just told this forever. But then when you actually sit and question it, I'm like, well, what? Why is this the way? So I really like talking about kind of the history of things too, or trying to highlight people that have kind of been forgotten in the history of, these breakthroughs and stuff. But it's also hard to not burden students, like we are always in a panic, about changes in the world and everything going on. And so sometimes it is hard trying to find that balance of like, I'm trying to spark curiosity and I'm also trying to not like worry people more than they already are. Right? But like, if it's something that's interesting, like it's okay to question it. It's okay to, get engaged with it, beyond whatever we're telling you. Like, if you're really interested, like, go do your own research, like, just start looking up articles, like, talk to folks that can help you, like, learn more about it. But going back to the question, I think, was the question actually asking, like, the breakdown of it? Because I feel like I would say I'm probably, like, 60 to 75% lecture, and then I'd say like 40 to 25 versus for hands-on stuff. But in my courses, I had a really big adjustment from my first semester to now, because in the first semester I was teaching 2 days a week 80-minute sections, and now I'm teaching 4 days a week, 50-minute. And so that was a really big adjustment. And so looking back on the fall, I wish I had done more of kind of those breakout sessions or activities. And so that's one thing I'm hoping to change in the future. And whatever classes I'm teaching is building in more of that time to do those sorts of things. But I know even you sometimes like have students prepare stuff ahead of time. So it's like sometimes I count that into like that non-lecture component. Like if I'm asking you to read a short article before you come to class, like to me that almost falls into that like non-lecture category because it's like beyond textbook material, but I'm still asking you to like read it.
Right. And that's I'm probably in the same range with the 60 to 75% depending on the material or the class. And that's kind of the tough part is and this is tough, any students listening, this is tough for all of your professors is, well, some of them don't put as much effort in and they're just going to read from it. I've heard of the big schools where they read from the textbook. We don't do that. But even with the lecture, it's hard because like when there's really hard stuff coming up, I feel very like nervous about not lecturing about it and putting it in class. I feel very nervous that that's not going to get the like selection it needs. But I know that I can't just do that all the time. So you really have to like call your shots as far as what you think is going to be the most pertinent information, like that you're going to be up there explaining, and hope that that crosses over so that when you build a bigger picture, those foundations and scaffolds are there. I definitely have talked with like some of my students, and I've always kind of broached the idea, like how much material, for example, could we flip the classroom with? Could I give you guys 2-3 videos that you would watch, and then we would like discuss and like do a couple things in class, and then, but would I know that you actually did them, unfortunately? And so that's like always the hard thing is like, as your professors, we would love if we never lectured and you guys would like, just like watch videos and read the textbook, but that's not the way that everybody learns either. That's the other thing is like, lecture does get sometimes a bad rap and it's like, oh, this is boring. It's still been working for quite a while. So there are very active dynamics of lecture that like make it a good delivery system. But I have had those like fun debates with some of my students, like, could you guys just take the test if we just like discussed implications and wrote all class? And some of them are like, yeah, definitely. And they're like, what are you talking about? Others are like, no, like, you need to explain mitochondria to me because they are complex, even though they're fun.
I think too, like, you're talking about like the foundations, like both of us are, a part of introductory classes, but also teaching like kind of upper level classes. And so kind of the difference, like I see more of kind of the non-lecture stuff for the upper classes, because by that point students have had those foundations in the previous courses. But I think the other thing too is I'm constantly having to remind myself of, oh yeah, I've been working with phylogenies for a decade, but for some people, this is the first time they're interacting with them, and so it's always great to slow your, passionate professor down, if they're going too fast, and it's because we're so interested and engaged in what we're doing, and, for us, we've gotten so comfortable talking about these things and teaching about them that sometimes it's hard for us to really step back and be like, oh yeah, if I was learning this for the first time or trying to put myself back in that kind of headspace helps me. But it's always important if you feel like, your professor's leaving everyone in the dust just because it's something that they've lectured on for 20 years. Like, slow them down. We're
not trying to sound smart. We're probably just excited in that case. And because that's what I did. I think I've done it before. Whenever I get the chance to talk about a cancer anecdote, I think I obliterate my classes every time. They're like, did he just say the cancer cell eats itself? Did he just say that macrophages get zombified by these? And what's a macrophage? I think that I always forget, like when I was hearing these things, I had like all the foundations set, and that's like when light bulbs started going off for me. So I always have to like keep careful track of, make sure that we are on track when it comes to what I'm about to talk about. And I always, like I said, that's why I think like evolution ecology might be some of my most straightforward stuff when I'm teaching it, even though it's not my field, because I'm like, this is exciting because this is exciting. Like I just learned this or relearned this and saw this again like a month ago, and I'm excited about it again. So we're going to be excited about that, not something super advanced. And that's why I always like wish that I could, I'm glad that we shared the office, because then I could ask you about stuff. Like I learned about George Foster Wallace from you, and I was like, oh my God, this poor guy. Like he like discovered everything. So So when we try to find that balance, we're not trying to force you to talk to your neighbor. We are literally trying to help your brain undergo something a little different, and that's good for the brain to see a situation in a new light. And, we always try and, I think both of our J terms did a good job, integrating like some ethics and conceptual questions as it related to the material, because that's, a 2-hour class or a 3-hour class is going to take some time. Maybe you do need some time to discuss and sit and think for a while. Those can be tough. So I think I also got a question from a couple of my students on how introductory labs are kind of done. And I had this group of students be like, can't you just trust us and we'll just like run experiments? And I was like, I obviously trust all of you, but there are definitely like certain things that it helps to have a couple repetitions with, like gathering data and then interpreting data. And I don't, and we're never wanting to like belittle you or pretending that you have training wheels on. Even if you are ready and you do feel like you are past certain stuff, then use this time to really get like an advanced handle and start thinking about, hey, how would I actually apply this to a question that I have, for example? How would I apply this in a scenario if I want to look into a certain protein that is associated with Alzheimer's? Would I, what kind of patients would I test? What kind of things could I do in a cell line, for example? I think 102, or at least the Gustavus version of 102, that's the first time that we put students in front of living things, and I always think that's really exciting. So I think that gets kind of that real stuff coming in, and I think...
Well, we had cockroaches in 101.
Oh my god, I forgot about the cockroaches. Yeah, so for everybody listening off context, for the Bio 101 class at Gustavus, you have to handle these hissing cockroaches as part of like a CO2 respiration lab. I gotta be 100% honest, like I've handled bugs before in some of my research. They were terrifying. So I went over to the pen and was like, okay, like I'm not gonna show you how to handle one of these, like you're gonna jump in and do it, students. That was purely because I was terrified of these things. And they hissed at me, and I think one of them I'm convinced bit me. I don't think, maybe not, but still, I was very upset. And so that was tough. I totally forgot that. I blacked that out, I guess. But trust us, when we do the biolabs in 101 and 102, those are exercises that I think, when it comes down to it, are very good examples of how much you put in is going to be how much you get out in a lot of cases. Some of them are tougher because some of them are simulations in a lot of cases, but that's okay. A lot of you maybe will get to a point where you are dealing with environments or cells or mice or patients someday, and you want to have practice. You want to be able to remember some training from when it was low stakes. Like I've told some of you, that's the big reason I don't work with mice, is that I make mistakes in lab, and I never want to have to be like, all right, these 8 mice, they're done. I never want to have to make that choice, and I never luckily have had to make that choice because I've kind of avoided making that choice, which is nice. Not everybody has that luxury, but remember that these training steps are not there to, put you down, basically. We're just, kind of showing you some ropes so that everybody emerges after, let's say, these 2 classes, and then jumps into something big like the cell and molecular class, or like 202 and EEB, for example, where you are going to get tasked with making an experiment, for example. You're going to get tasked with finding that data, asking a question, finding data, making a conclusion, and interpreting what that conclusion means. And I think that Gustavus is a pretty good fast track when it comes to, getting students on that path. I think even as a freshman at Carleton, I think the labs were really obviously super well done. I don't know if personally I was ready to be in such an advanced lab yet, which is so bad that I'm a biology professor now. But I definitely don't think that I personally had that level. I don't know if maturity is the right word, but I definitely didn't appreciate how much work went into those labs when I was an undergrad. That's another thing that professors love doing. When I was in school, quote, I should have taken advantage of all these things, so you guys should be studying and being involved. Don't go watch Netflix, please, basically. But, we have that luxury of, wanting you to do that now, and we remember how hard college is, trust me. There's a lot flying around, so it's okay. You go ahead.
I think one of the other things, too, that maybe, and again, every school is going to be different, but at least at Gustavus, people start out with 101, but doesn't mean they're going to go to 102. Like, some people went down different routes. And so I think 101 is used as a way to, like, build confidence, you know, especially for folks that haven't, you know, depending on what school system you were a part of, like, some of these tools and equipment, like, you might not have used before, you know. And so for those students that have had that experience and feel like, oh, it's so slow or all that, like, take it as a time to also kind of help your fellow classmates too, you know, and be able to help bring them up and build their confidence with, you know, these tools or whatever equipment is being used that they might not have had experience with if you had, instead of viewing it as a, like, oh, can't we just, you know, run off, you know, like, we want to be inclusive to everyone. And I think especially for me, that was important because I was the first person in my family to go to college, and I, you know, at least in my high school, like, I had biology in, how was it, 9th grade? And then biology was 10th grade. And then I didn't have it until my sophomore year at Gustavus. And so I think that's another thing with biology is you have students coming in that maybe just had AP Bio, but then there are some students that haven't had it for a couple of years, or their last bio class was, like, human anatomy in college or something, or in high school, sorry. And so there's just such a wide range. I think it's our chance, you know, at the institutional level to try and get everyone kind of situated to be able to set up for success, you know, in whatever next course they're in.
Right.
But I do feel like sometimes there's so much, like, equipment that's thrown in that it seems a little overwhelming, you know, to keep track of whatever sensors and stuff. And so sometimes I view their approach less is more because, you know, in the future labs you can build on that, but it's really important to get the solid foundation first before you throw everything in the kitchen sink at students.
I remember, I think me and you would probably be an undergrad when they had the spectrophotometers that were just, like, so bad to work with. And that's how so many of our labs were, with spectrophotometers that were just, like, the worst things on the planet. Like, if the thing was a little filmy, it wouldn't read. If you put it in the wrong way, it would break. I just remember at Carleton, whenever I saw the spectrophotometers come out, I was just like, oh my god, today's going to be so bad.
And I think that's the hard part too, is, like, it's immediately putting up these kind of walls for students of, like, oh my gosh, it's too hard. I can't figure out this piece of equipment. But it's like, you could be understanding everything else in the process, and you could explain it to me, and I know you're on board, and it's not your fault that the machine's not working properly. And so I think that's my frustration too, having all of these, you know, oodles of fancy, you know, tools to use sometimes, is it kind of muddles the confidence or the ability that students might feel that they have for things just because they're not able to get, you know, this machine to work. And, oh, if I can't get this machine to work, then, like, I just need to not be a bio major.
Right.
Like, that's not true.
Because it's weird, because it, like, yeah, because you get better at it in grad school. Like, I was actually super afraid of lab in grad school. It's like, all right, here we go. And then it was like, okay, like, you're going to need to learn these techniques. And, like, that is usually, like, a scary, like, 48 hours. Like, when you're about to perform your first technique on something.
Yeah, I was doing, like, the phenylchloroform, like, extractions. That was terrifying.
But yeah, so I think we had a couple other questions, but I think let's focus on the last ones, and because it's good, because I think I talked so much. You guys could speed through this at 2x motion, and you could still hear my voice pretty much the same. So, let's see. There's kind of two questions that are connected. How did you become a professor? And remember, we're just visiting, so we're not, like, real professors yet.
Yeah.
But that's okay.
We're not real.
Yeah, we're just, like, kind of, like, fake, like, pretend professors. You still pay full tuition for us, though. How do you best prepare, and then how do you best prepare as a professor, especially in this first year? And then we'll address the final one after that.
So first question was how did we come to where we are now? So I've always enjoyed education. I've had a lot of years of experience with environmental education, and then as a part of my graduate program, I also created and ran an outreach event series for science education, connecting researchers and businesses. So I would have researchers come to the businesses at night and share their research, and students and community members and their colleagues would come listen. And so I ran that for 4 years. And so for me, like, I really like connecting resources and getting people interested and engaged with things. And so I was like, I know I'm interested in teaching, and I'm still trying to find where that balance is of exactly what sort of role do I want as an educator. But honestly, for me, like this position, I was like really scared to apply, and I was like, oh, I don't know, but I wanted to put my name in, and so it's been really nice to come back and be teaching classes that I took. Number one, I have a lot of my binders from when I took the classes.
That's
so
awesome.
like I have my exams I took. So you know, again, things have changed in the last 10 years or 13 years or whatever, but I think just seeking out the opportunities that get you really excited. And this was one, just because my, you know, past connection. But even just seeing other job postings too, just picking out the parts that really are interesting to me that are education related is exciting.
Yeah.
To kind of see what possibilities exist.
Oh, that's awesome. And I think I was kind of like a different student. Like I've talked with some of my students about this, how I'm very bad at absorbing content in the immediate future. And so Carleton's on trimesters, so the classes happen in 10 weeks. That's not actually a recipe for success, unfortunately. So as much as I would love like talking about stuff and was very still interested for like years after classes, I was always like, shoot, I didn't really apply that on the exam as much as I wanted to, even though now I like am very interested in these ideas still. So for my path as far, I think it was two main classes that really, like when light bulbs started going off, I was like, oh my gosh, like there's this entire world to still be opened up in biology that I'm just always excited about. One was my genetics professor at Carleton, Stefan Zweifel, and then another was Debbie Walzer-Kuntz at Carleton, and she does science education and virology. And in both those classes, Stefan was very good at, I don't know if he was tricking us or just making us like wowing us with discussion, and like he would like pull the sheet from under us and be like, did you actually know that like this is how holiday junction in DNA looks, or did you actually know that like certain things can get controlled by other genes in this manner? And I would always just be like, there's so much more to this, isn't there? Like he has a whole like valley of stuff we could talk about. This stuff, it's so exciting. But I remember one specifically in a seminar class on virology that Debbie was teaching, and we were all in groups studying HIV primary lit papers. And I remember looking up with my group, and I kind of looked at them and I was like, are we all thinking the same thing? And we looked at Debbie and it's like, it was the first time that the papers were starting to come out that said, with this combination of retroviral drugs, a patient can exist with HIV and they're not going to die. And that was the first time, like, we all looked at Debbie and were like, is this person winning? Like, this isn't a death sentence anymore. And she's like, that's how we win. And we were all just floored. And I was lucky that, like, that's what we were reading about at the time. I remember the paper was from 2011 and it was 2012 that year. And I was just like, oh my gosh, this is so exciting. This is so interesting. And to this day, I'm still so interested in viruses. Not currently, unfortunately. Now I'm just upset that we get canceled our year. But everybody should stay safe. But I remember with education, I always thought, like, I want to do what they do. Like, that was awesome. And have those, like, kind of those connections with students and, like, seeing what means something to students and how you can connect that to something exciting. I had really long drives back from Carleton back to Colorado in between all my school breaks. And my dad and I were talking on one of these drives because it's super late and we're just like, you know, mulling about stuff. And he's like, so what are you going to do? And I was like, well, like, I kind of want to go to med school, kind of, maybe, or I really wanted to be a veterinarian actually, but I graduated without any animal experience. So that was a big thumbs down. And then he's like, well, what about, like, if you studied biology more? Like, you really are so excited on these drives and you tell me everything that you studied throughout the term. And, like, it always seems like you get more and more excited and more things connect. And he's not a science person. And so it was super fun. And I was like, you know, if I'm going to write, like, a personal statement for one of those three things, this is probably going to be the easiest one to write. And that's still what I tell my students is, what would be the easiest personal statement that makes you unique for this type of, like, you know, school, career, anything. And then I always ask him this question, and maybe this is mean, but would you do that profession for, let's say, $20,000 a year? And if some of them for medicine, they're like, hey, no. And I was like, well, then remember, like, there has to be that big mega motivation for those years when you're in residency or you're, you know, struggling through a library and you're just like, oh my God, like, you know, that motivation has to be very intrinsic. And hopefully what our job is as professors is to help you find that. And, you know, I had professors that helped me find that. And so someday, maybe I can help some people along the way. And that was fun. I definitely still really miss a lot of the times with my research sometimes, like obviously that I don't directly do. And I think because it's very medical associated, I do know that a return to that's possible someday, because I feel like I could still have that effect there. So we'll see how things shake out this year. That's still up in the air for all my normal listeners. We'll see if this becomes more regular again if I'm not teaching.
Yeah, and I think, like you're saying, you want to kind of give back, and that's something I think about too, is like I had really influential mentors, and I saw, the ability they had to communicate these really cool things to people and get people interested and cared, but also knowing that they cared about me, I think was a really big part, and so I take mentorship really seriously, like you do as well, and so I think that's another reason why I like these sorts of education roles, is because you are able to kind of make people see that they're capable of a lot more than they think they are. Because I always think sometimes that, like, I should have been someone probably that didn't make it and did drop out. Or, I'm not an A student. I struggled in school, but that struggle helped me, become more resilient. And so I never want students to think, oh, just because I'm not getting every single A possible, it doesn't mean you can't, continue down that path.
No, definitely.
But again, your background in, like, medical sort of related things, maybe sometimes it is a little bit more pressured, but I just want people to know that it is okay to, quote unquote, fail a little bit along the way as you're figuring it out, and have that kind of intrinsic motivation, like you're saying, is really important.
No, and I think that's always good to, like, have those experiences where you kind of know what it's like to lose and get back up again. Because you don't always get that through academics sometimes, where it's like, sometimes you can't really pick yourself up in the same way, or at least you haven't learned how. And like, and it's also hard for me to even say that you learn how to not give up. I don't know if you can even put that something so powerful towards how to just learn that. I know that I had supports systems in the same way that I mean, I've never really admitted this, but I definitely struggled at Carleton. There was a lot of times where I was like, this place beat me. I remember I talked to my parents about that. I was like, it's too much. Like, I'm falling behind. I'm this. And so I always try and remember that with our students, and just remember, like, you can get through this, like, if you believe in it, because we believe in you. And I remember going back to class that Monday after, like, a hard weekend or something, and being like, they're still here. They're not going to give up on me, and neither should I. So that was exciting. Dang, yeah. Yeah, school's tough. We don't forget that. Trust us. And graduate school's tough, too. You said, hey...
It's still tough for us.
Yeah,
on the other side of things, it's still tough.
So as far as how we prepared, though, at a med center, it was weird. I didn't have any undergrads to teach. I had to, like, go email people in departments and be like, can I teach one of your classes, please? And so I gradually, like, piece by piece, started building a little bit of this teaching resume, and then... Larissa Wolfenbarger at UNO offered me to teach microbiology, and I was like, I've never taught microbiology, but I'm so thrilled. And I learned microbiology in a month, and we taught it, and we had a great class. And but your resume with teaching is so much, like, bigger, because you, like, started an organization and stuff. I should have been doing that, but one of the things that we've talked about, I've shared with you, is that I did have a research mentor that was not very thrilled about teaching, and we had conversations about that we never came to an agreement on, and that's okay, and I respect him a ton, and I learned tons from him. Probably one of the smartest people I've ever met to this day, possibly the smartest, and it's, but it's still something that we could never really agree on about priorities as a grad student, and I understand where he's coming from, given that he was mentoring me to be a scientist, and I was, still very interested in teaching, and so that was definitely a different dynamic, to say the least, sometimes. But
I think with, like, the actual decisions of, like, preparing material and, like, kind of organization of a class, it's kind of, I don't know if students know this, but generally, like, if you're teaching, like, a core course, it's not just one professor, there's multiple, so there's kind of behind the scenes things that happen, prior to the semester starting, where there's, like, a certain order of things, that we're covering, and we kind of have to have some consistency as to what's going on, but then there is flexibility too. And so something I always try to do, and maybe my students will disagree or tell me that I'm failing at this, but I never want them to leave my class being like, well, that was a waste. Like, what was that for? What did I learn? Like, I want there to be at least, 1 or 2 or 3 tangible things that they're learning that are actually like applicable to either things that they've noticed in the world around them previously, and now they're like, aha, now I know like what that actually, like why that is the way it is, or something that gets them interested in something that they want to like talk about with their friends with. That's like to me a huge success as an educator. But I think, all the experiences that I had, it's like I want people to know it's not like we just land in these positions. I graduated in 2010, so it's like 10 years of things led me to this point now, and that had to do with like, I did 2 years of AmeriCorps, getting a master's degree, starting my PhD, so it's like, there's steps along the way, but all of those steps are also full of amazing opportunities that you get that maybe in the moment you're not realizing how important they're going to be, but looking back, everything makes way more sense in kind of line of, as you're trekking through life and the different jobs you're having, it's like it makes way more sense looking backwards than if someone were to tell you these are the steps you're going to take.
Oh, I know, that was definitely like kind of the fate of that. So like Larissa was the one that offered me the teaching position for intro and then micro at UNO, and that probably happened because I took a class that a colleague recommended at UNO that was a teaching class, and then she was leading it. And then one day she was like, hey, you're like a PhD candidate, so you can technically teach this. Would you be interested? And I was like, oh my God, yes. Like, this is so exciting. We finally did it. So you're right though. Cause that makes me think of that, like that moment, cause I definitely wasn't thinking of taking that class. It was not high on my priority. I was like, I'm going to graduate in a year. I've already taken a teaching class, but I learned a ton again. And so that goes to show we're always right. We're always making you learn stuff and never wasting time. But yeah, whenever we prepare, it's very collaborative. I would say that not only the two of us work together, but we were very quick to go to some of the senior faculty and say, here are the strategies we're using. What do you think? And because I agree that I always tried to make that class worth what students are paying for at Gustavus, because I've seen what students pay for at large universities where you're not getting a lot of attention and there's not a lot of care behind those classes. And I'm hoping that, obviously as I get better as an educator, or, maybe someday as a scientist, we'll see that that effort is that effort's always going to be there, and that there are going to be tangible results that you got something out of the class and that it was valuable. So we're always trying for that. If you do have a professor that's not trying for that, then that's a problem, I'd say. I don't like that. But I mean, again, there's always different styles though too. So maybe that's what it is. But awesome.
But I think too, when we're like deciding topics, it's like obviously if we're gonna kind of gravitate towards the things that really interest us and excite us. So sometimes like the articles I have people read or the videos or whatever seem maybe very out there sometimes, but I'm hoping they're able to kind of wrap their head around like, what is the connection to class and why the heck is Katie asking me to watch this? Because it's really exciting for us to be able to, teach about and, learn with them, because I feel like I'm always learning too. And so I think it's good for students to know that, it is a collaborative process, even if it doesn't seem that way. Like if I get good feedback from you on the activities and the topics, then that helps me see that, you're liking it and learning from it, and I'll keep using it. But if you're telling me that, oh, this really didn't work, then it's good for me to also take that in. And Keenan, I'm sure you get that sort of kind of feedback from students too. It's important. We want to be doing a good job. Like we care. Yeah,
definitely come up to either of us and it was like, this isn't working. Yeah, don't be afraid of ever coming up to your professors and being like, this isn't working so much for me, or like, I didn't understand that part. We are excited to help. Like, trust us. That's not going to happen in every grad school or clinician or anything. Yeah, sometimes people are impatient. I get that. You're at Gustavus, though. You're at a smaller school where, like, that's what we are excited to do every day. That's, like, the best part of our day is when we can talk with students, honestly. And that's why...
the last 2 weeks have been so hard, not seeing our students, like, and not even interacting with them or anything.
Sucks.
Like, it's weird. We get to know you and we care about you, and so it's, like, it's hard,
I was having so much fun with both of my 102 labs because I'm not lecturing in 102. So it's kind of just been fun to be like, what are we going to do this week? Like, this is going to be so cool. And like, there were very little consequences for me just being like, this is lab, hooray. Like, and so, and it's obviously going to be okay. Like with everything, like I love the tree project. Like, I think it's great. I think it's hilarious that you chose a dead tree when you were
I know, yeah. I need to email my students and just be like, make sure you pick a live tree for the phenology report. Because I did not.
I do have a couple students that are not in an area with deciduous buds. And so I have one in my backyard, so I'm just going to take like pictures and email them every day and be like, yeah, I'm going to be like 4/2/2020 tree,
Yeah,
large
here it is.
picture, and then like some buds. And
Yeah.
you're gonna be able to measure it and get observable data, so that'll be good. So we'll see. But no, thanks so much for talking with us, or talking with me, Katie. I don't know if it's us. I don't know if anybody listens to this podcast anymore. We'll see what happens when we light up the listens. We'll see. And but we just, yeah, this year teaching has been amazing, and it's fun, and like we love what we do, and that's so know that you're in a place where you have professors that are very excited about coming to work every day. Like it's very hardly work for me. It's very much how I feel when I'm doing like clinical lab science, where it's like, this is so exciting. This is real difference making stuff. But at the end of the day, like I love talking with students, like it's the best. Yeah. So.
And I think too, because it's both of our first years, like there is a little bit of that, like students feeling like guinea pigs, I'm sure. And they are, we're learning. This is our first attempt at everything. And so, yeah, next year I'll do some things a little bit differently. And 5 years from now, wherever I end up, like I'll maybe do things differently too. And so, I think that's one of the things about the workload this year was big. We're kind of
we were busy
restructuring things and making it.
We were, I remember, my wife commented that one thing Carleton prepares you for is like you are never not working at Carleton because of the trimesters basically. And I remember talking to my wife Emma and was just like, this is like what it feels like. I haven't felt like this since Carleton, where it's like it's never stopping, like the work. But it's obviously awesome because it's all from a point of like, I just want to make this better. I want to make something new that's going to be more exciting or more engaging, and we're going to learn more, and we're going to be more fulfilled in that case. So that would be awesome. So yeah, it's awesome.
Well, thank you, Keenan. I meant to ask earlier, so I do have a Twitter account, so I don't know if you post this on your Twitter. I don't know where
I will actually,
this,
I haven't used that yet. Yeah.
Tag me, I don't know what it is.
You should retweet me. I'll have more than like 3 followers. I think my cousin and you follow me, and like another person I know from Mayo, so we'll maybe get that out.
So if folks do want to contact me, they can contact me on Twitter, so if they look for that.
Okay. And I'll, I think... You search for me on Twitter. It's just my dumb picture, and there's only one Keenan Harder in the world, so you'll find it. And that's fine. And I'll post Scout pictures of my Pomeranian Scout, and you guys can meet Scout for the first time. She is beautiful. I did have a molecular biology student, one of the advanced students. He did his journal club on the Merle phenotype in dogs and how like the melanin gene in dogs gets disrupted by an intron that gets interrupted by a little transposable element that inserts itself too many times, and that's how it's like messes with the amount of genes, and so that's why Scout looks like a little patchy mud ball. And so don't tell Scout that I said that. But
yeah, she'll be offended.
I know, she does get she's so happy about quarantine, trust me. Everybody's talking about their pets, like, what are you still doing here? Scout's like, this is great.
Well,
like,
see, I have a cat, and I feel like I'm disturbing her, like, napping routine for, the 20 hours of the day, but...
Wow, she'll be good.
Cool.
Well, again, thank you so much,
thank
Katie.
you.
Yeah, this was super fun.
Thank you for the questions.
No, yeah, and thank you, students for questions. Yeah, this was fun. I wish we had more. We could do it again if we end up with more. So actually, yeah, if you have, and I'll send this in the email to follow up. If you have any follow-up questions, like we're always glad to do another, or just kind of post some answers and have a Q&A. But in any case, thanks so much for listening, and this concludes a very special, fun episode. Thanks for listening. Bye.